Hi everyone, my name is Emily and I'm going to talk to you today about my work on the links between spatial distribution of food and perceived sperm competition risk. All of this work could not have happened without these three people, so I'm going to start my presentation today by thanking them, as they deserve credit too. So thank you to my two supervisors, Mike and John, and to our amazing technician, Fliss. First off, I'm going to give you a little bit of background information on the history of sperm competition. Sperm competition occurs when two or more males' sperm compete to fertilise the same female's egg. Males can detect when rivals are nearby, so they know that if they mate with a female and a rival is around, that it is likely that another male will also mate with her and that their sperm will have to compete. So they have a perception of the risk of sperm competition. We can alter perceived sperm competition risk by manipulating the population density. And if we were to do this, we would expect to see changes both short-term, from an individual perspective, behaviourally and physiologically, but also more long-term evolutionary changes too. This was first discovered back in 1970 by Jeff Parker using yellow dung flies. Back then, researchers were only interested in interspecific differences, but it soon became clear that plasticity should benefit individual males. And this has indeed been shown in a vast range of species, including other invertebrates, fish, and rodents. Drosophila melanogaster are a popular model organism, so it is not surprising that we know a lot about their responses to varying perceived sperm competition risk. Responses found include increased mating durations, an increased number of sperm, both amount produced and amount successfully transferred to females in mating, and also a number of changes to the composition of accessory proteins found in seminal fluid, all of which lead to an increased fitness for the individual male. This effect of increased mating duration has been shown repeatedly throughout literature and is therefore a well-known response. It is what we use in this study to find any possible responses to perceived sperm competition risk. Despite these important fitness effects and the vast number of studies completed, most of these results have been found by directly manipulating the numbers of co-housed males, with the most common treatments comparing solitary and paired males. But solitary versus paired is not really biologically meaningful. It would be rare for an individual to spend its whole life in solitude, or even none of its life in solitude. So why do we see these results in the lab? We don't yet know what mechanism could be driving this in natural populations. Density in natural populations is more flexible. As you can see from the image, you'll find clusters of flies in the wild, and then also areas where they are more sparse, but rarely, if ever, solitude for its whole lifespan. Considering this, there has been little research into a more biologically relevant stimulus that could alter perceived density or perceived sperm competition risk. So this got us thinking, and we wondered whether varying food resource distribution could alter perceived sperm competition risk via a changing social environment and the density of the local population that the male perceives himself to be in. We further split this question into two parts. How does varying food resource spatial distributions, the physical environment, affect the social environment? And how does this in turn impact male mating behaviour responses? To test this, we used three treatments: clustered and dispersed distributions of 4 different food patches, and a control, an even coverage of food. We added 4 males to each arena, and they remained in these treatments for 3 days. Images of the individual positions were taken every 15 minutes, which enabled us to calculate Euclidean distances, of which there were 6 in total. Here's an example arena where you can see these 6 distances. We then use these values to work out the mean individual distances from the 3 rivals for each image. After the 3 days in the treatment, the males were paired with a standard 7-day-old female, and we observed the time taken for him to begin courting and mating, and also the duration of any successful matings. So what did we find? How does varying the spatial distribution of food resources impact the social environment? In this graph, the top line in yellow is individuals from the dispersed treatment, black is control, and then the lower green line is those exposed to clustered food resources. As you can see, the difference between the observed mean distances between individuals across the 3 treatments is significant. And by the 3rd day, the distances between individuals appear to plateau. And we found that males housed on clustered treatments were twice as close to rival males as those that were housed on dispersed resources. Now, before I move on to what this means, it's worth highlighting this interesting behaviour taking place on the 1st day, where the opposite actually appears to be true, because I'm going to come back to that a little bit later. But we therefore now know that altering the distribution does, as expected, influence social environment. Encounter rate and the chance of individuals meeting rivals increases when resources are closer. But now on to the second question. How does this then impact responses in male mating behaviours? We found that mating duration was longer in males on clustered food treatments in closer proximity to rivals when compared to those further from rivals on dispersed food patches. The eagle-eyed of you may have noticed these 2 individual points, and we were worried that they alone were causing this result, so we retested this excluding those values, and the difference remained significant. So we are confident in this result. Males found in closer proximity to rivals mated for over 2 minutes longer than those on dispersed patches. This is evidence that spatial distribution of food resources influences perceived sperm competition risk. And this is actually, to our knowledge, the first study to find this effect of increased mating duration without changing the actual density of individuals in each treatment. So now we have the answer to our question. Yes, varying food distribution does alter perceived sperm competition risk. And this means that varying patchiness could be one mechanism that is driving responses to perceived sperm competition in wild living populations. But what do we do with this new information? Well, we would like to try and better understand this potential mechanism by testing the theory in wild populations. We'd also like to track individuals so we can better link individual responses. Now, I said I was going to come back to this result. Tracking individuals would also help us to understand these intriguing dynamics taking place in the early stages of the treatment. This effect does not match what we expected to see, that males were defending individual patches. Inspection of photographs suggests that males initially cluster together away from food, before sorting themselves into individual territories focused around each patch. But it is not yet clear what is driving this initial clustering behaviour. Tracking individuals would enable us to explore this further. Unfortunately, as is very common at the moment, our plans haven't quite worked out as we hoped, as we have not been able to get into the lab or out to the field to complete any of this research yet, and I am now rapidly nearing the end of my PhD. So for now, if you would like any more information on this, you can read all about it in our preprint on bioRxiv, or ask me any questions in the Q&A session. Thank you for listening and learning about patchiness and sperm competition risk. I'm looking forward to meeting you all soon.